So now we're going to go ahead and look at the actual process of meiosis. And we're going to, remember, keep count or keep track of some of the terms that we used before, like haploid and diploid, and looking as they come up through the process of meiosis, through the process of dividing the cells to form gametes, or sperm and egg cells. So these sperm and egg cells, that's the goal here of meiosis, is to make cells to pass on genes to the next generation through sexual reproduction. Now remember, we did talk about mitosis. Some differences here. Mitosis makes copies of cells. They don't make gametes. It makes copies, exact genetic copies of cells, and that's for growth and repair and development of tissues. Meiosis, when we look, remember, its sole purpose is to make sperm cells or egg cells. And it's happening where? It's happening in the gametes, in the ovaries, or the testes. Only location for meiosis in our bodies. So the whole purpose, remember, of meiosis is to reduce that number of chromosomes from 46 down to 23. Okay? And we want to do that so that you can combine an egg plus a sperm, right? Here, egg plus sperm, to make what we're going to call a zygote, okay, which is a fertilized egg with 46 chromosomes. So in order to do that, we have to take cells that are starting off as what we would call diploid cells, and we have to turn them into haploid cells, cells that have half. So these cells have 2 copies of each chromosome. These cells down here, when they're haploid, have just 1 copy of each chromosome. And in doing that, that's so that when they unite, when they meet up here at fertilization, you're making a zygote which has the proper number of chromosomes for that cell. And so we're going to go through the stages of meiosis, which have a lot of overlap with the stages of mitosis. We're going to see prophase, metaphase, anaphase, telophase all over again. But we're going to have a couple of unique structures and concepts along the way. First up for our phases of meiosis is going to be interphase. This is the time in between division. During interphase, the cell is going to replicate its chromosome, so it's going to copy the DNA. Okay? So you're going to go from that having just a single copy of each chromosome to have 2 copies of each chromosome. And so you will have 2 identical sister chromatids held together by a centromere. We talked about you're going to have basically a mom copy of a chromosome and a dad copy of each chromosome. That's going to be during interphase. During prophase 1, you're going to see just like in mitosis, you're going to lose the nucleus, so no nucleus. No nuclear membrane, okay? The chromosomes coil up, and the unique structure here is something called a tetrad, okay? Now, we talked about chromosomes are homologous, okay? So homologous chromosome, remember, copy from your mom, copy from your dad. They have the same genes. Let's say they all have a gene A on them, okay? And those chromosomes are homologous. Now, what a tetrad is when you have homologous chromosomes that line up on top of one another, okay? So tetrad, think 4, okay? And these chromosomes are lined up overlapping. Their arms are actually intertwined or wrapped around one another. So that's a key hint as to whether a cell's in prophase 1 or prophase 2, look for the tetrads. If there's tetrads there, it's got to be in meiosis 1, okay? So we also see same things that we saw in mitosis, spindle fibers and centrioles appearing. And so we'll come back to this, but these tetrads actually pair so tightly that the arms can get crossed and attached to one another, and something called crossing over can happen, which is where pieces of chromosomes are traded back and forth. Crossing over can only happen when the tetrads are in place, which is going to be prophase 1 and our next phase, metaphase 1 of meiosis 1. And so in metaphase 1, we're still going to use that hint word middle, okay? But in metaphase 1, what lines up down the middle are the homologous pairs line up down the middle. So you get the tetrads. And so what you have is basically like the dad chromosome on the top and the mom chromosome on the bottom side. And what's going to happen is when the spindle fibers contract, it's going to pull those whole chromosomes, the whole X-shaped structure, towards opposite sides of the cell. And so this is a look at crossing over. And so we're going to look at a couple things here. Here's an electron microscope picture of crossing over happening between a tetrad. And basically what we're looking at is that the legs or arms of these chromosomes can get so close and so wrapped up around another that they can actually trade pieces. And so what that essentially means is normally you would have, you know, your dad's chromosome in red and your mom's chromosome in blue. And after crossing over, what you could actually have is your mom's chromosomes here, your dad's chromosomes there, but your mom's chromosome actually has a little piece of your dad's genes on one side, and your dad's chromosome has a little piece of your mom's genes on the other side, okay? And so what this does is it separates traits. So like if eye color, let's pretend like eye color is up at the top, hair color is at the bottom, now you have chromosomes with mixes of your mom and dad's eye and hair color through this process of crossing over. This is one of the things that makes variation in sexual reproduction. Anaphase 1 then, we're still going to go with apart, right? This time it's the homologous pairs that are pulled apart, right? And so they're moving towards opposite sides of the cell. They're getting pulled in opposite directions. Notice crossing over has happened. And the goal here is to send one copy of chromosome number 1 to the top, one copy of number 1 to the bottom, one copy of number 2 to the top, one copy of number 2 to the bottom, so that basically each side of the cell gets one copy of each chromosome. Now, in anaphase, we actually have something called random assortment happening. What that means is it's a mix of maternal mother and paternal father's chromosomes that go in either direction. In other words, this one could be your mom's and that one could be your dad's, right? There's a mix. Now in humans, we have 23 chromosomes. So you can imagine you could everything happening from all 23 of your dads going up and 23 of your moms going down, to an even like 11 of your dads, 12 of your moms, and 11 of your, or 12 of your moms and 11 of your dads going in either direction. And it's random assortment. It's like shuffling a deck of cards there. During telophase 2, usually you'll get new nuclei forming, okay, right here around the chromosomes, and the cytoplasm is split in half in cytokinesis. It's usually lumped in under telophase here in meiosis, and you get 2 cells, okay, and they're actually now haploid. They have half the material that they should. That's a hard concept to think about. What you need to realize is they should have, right, when they started off, they had like 1 copy of your dad's and 1 copy of your mom's in each cell, okay? What they have now is not that. What they have now is they've got basically like 2 copies of your mom in one cell and 2 copies of your dad's in the other cell. Those aren't unique pieces of information. They've got 2 of the same chromosome, so they're haploid. They have half. They should have 2 full X's. They've got just 1 X now for each homologous pair, right? They got 1 chromosome from the homologous pair. They have half the material they should. So we now call the cells haploid at the end of telophase 1. So just as a quick overview, the DNA was copied during interphase, right? Prophase, the chromosomes coiled up, the nucleus disappears. Metaphase, the homologous pairs line up. Okay, either side, mom's on one side, dad's on the other side. And then anaphase and telophase, they move towards opposite sides of the cell, and we get two cells. So we've turned one cell into two haploid cells at this point. Now we're not done yet. We're actually going to go through a whole nother round of division that we call meiosis 2. And basically we need to break apart these sister chromatids, okay? Because if these cells went on to fertilize, you'd still get more than 46 chromosomes. You'd have 92 chromosomes, and that's not what we want. So we've got to break apart those sister chromatids. In order to do that, we have another round of division, okay? Now, this round of division doesn't have homologous chromosomes. It can't, because they should have been separated during meiosis 1, okay? And it should look exactly like mitosis, okay? It looks like mitosis, but happening in two cells. So it looks like mitosis in two cells at the same time, okay? And so we're gonna follow through there. So prophase 2, nuclear membrane dissolves, so that wears away. Chromosomes may or may not be coiled up. We see centrioles and spindle fibers moving out towards opposite sides of the cell. The cells are, what's our P word here? They're preparing, right? The cells prepare to begin the process of division. Okay, now during metaphase 2, we're actually seeing that the cells, it's the sister chromatids that line up down the middle. This looks just like it did in mitosis. And what we're going to see here is that these sister chromatids are eventually going to get pulled apart, but this looks like mitosis, just happening in two cells at the same time. We can still use our hint word here, middle, because that's where they're lined up. Anaphase 2, those sister chromatids are pulled apart. Notice that in this drawing, they're actually showing in a couple of places that crossing over had occurred. See these different colored ends on these chromosomes? They're trying to show you that the cells crossed over back during prophase and metaphase 1. And now you're going to end up with cells that have unique combinations of chromosomes that weren't there before sexual reproduction. In other words, unique combinations have been made through the process of meiosis. Telophase 2, we're actually going to make not 2, but 4 nuclei. Look, we have 4 cells here, so we've got 1, 2, 3, all right, and 4. 1, 2, 3, and 4 nuclei reforming. The cells are still haploid at this point, meaning they have half that amount of genetic material. And this is going to be basically the final end for meiosis. These cells are almost what you would call mature sperm cells or egg cells. And so meiosis 2, we're starting off with haploid cells, and we're ending up with haploid cells. So the amount of unique genetic information hasn't really changed here during this process, but we've split these sister chromatids apart, and we've ended up with 4 unique cells. Thanks to crossing over and random assortment, these cells could have some DNA from the mom and some DNA from the dad in them. So it's not like all of the dad's DNA gets passed on or all of the mom's DNA gets passed on. The offspring receives a mix of maternal and paternal DNA from both sides. So both the sperm and the egg are carrying a mix of DNA from that individual's parents. So at the end of meiosis, we took one cell and we turned it into 1, 2, 3, 4. We went from what we call diploid, having 2 copies of each chromosome, to haploid, having only 1 copy of each chromosome in the cell. Okay, these cells are now going to be what we call gametes. Okay, either sperm or egg, and they're going to be responsible for passing on genes to the next generation. A couple of unique terms that come along when we're looking at these processes. First up is this spermatogenesis, okay? And genesis basically means creation, and sperm is obviously sperm, and so this is the creation of the sperm, which occurs in the testes, and it makes 4 new sperm cells per every cell that starts the process. Now that's what happens in males. In females, we call it oogenesis, okay? The prefix oo is equates to egg. You know, we'll see terms like ova, which means egg, or ovum, or oocyte. So oogenesis is egg formation. In humans, we have a unique sort of way of forming eggs. We actually only make 1 egg per meiosis, and 3 of these tiny little cells that we call polar bodies, which break up. They cannot be fertilized. They don't turn into offspring. You make 1 mature egg in humans through oogenesis. Now, the great thing about sexual reproduction is it produces variation. Okay, so these are differences among the individuals between the parents and the offspring, and variations are resulting from a lot of things, but crossing over and recombination, along with combining genes from 2 parents, make variation in the offspring. So here's just a look at that process. Here's spermatogenesis. Remember, this would be in males as it goes through. Meiosis 1 would be the first round of division. So from here, basically to right here, meiosis 1. Meiosis 2 would be the second round of division to form the 4 cells. Now, when we look in oogenesis in females, what you'll notice is that it's an unequal division of the size of the cell. And the strategy here is that females, you want a big egg. You want a big egg because it carries all the materials, equals lots of materials for forming that offspring. Okay? The sperm doesn't really bring anything along. And so what happens is every time the cell divides, it sends a lot more like cytoplasm and organelles in one direction. All right? And so we end up with 1 really large cell over here at the ovum, and 3 of these smaller cells, which didn't really get very much cytoplasm, those are going to be the polar bodies. And the polar bodies just sort of, they go away. They can't be fertilized. And we end up with 1 large mature egg cell. So to kind of, zoom in or look at some things here, meiosis 1, this is going from diploid to haploid cells. Key feature of meiosis 1 are the presence of the tetrads, right? Over here we can see them lining up next to each other, the homologous pairs of chromosomes. Key process that happens here is crossing over and random assortment. Okay, both can happen here in meiosis 1 to produce unique variations or differences among the offspring. Meiosis 2 has no homologous pairs, and so it can't have any tetrads. So if you see a stage of meiosis and there are no tetrads present, look at how they're lined up down the middle, not in tetrads. That's a good hint to you that it's meiosis 2. And here we're going from 2 haploid cells to 4 haploid cells, and we're seeing all the spindle fibers are there, the sister chromatids are being pulled apart, all the stuff that we talked about in mitosis. So we'll do a comparison, but you're looking at genetically identical versus different. Okay, you have 1 round of division, 1 division for mitosis. You have 2 sets of division for meiosis. Okay, these are the key differences, and the presence of crossing over in meiosis. So these are the terms that just associate with the formation of those unique structures or or unique features that are present or found only in sexual reproduction. Genetic recombination, this is an idea that sexual reproduction makes variation, and that's sort of the whole point here of organisms reproducing sexually, so that you make offspring that are different from the parents, that might be better suited to the environment, that might be better adapted. Now we have a couple ways of doing this. First up, we've already looked at a little bit, random assortment. And this is during anaphase 1. That's when the homologous chromosomes move randomly to opposite sides, so not all the chromosomes from one parent go in one direction. Okay? It's like shuffling a deck of cards. So if you've got, you know, like dad and mom homologous pair, and then dad and mom homologous pair, maybe this dad goes up and that dad chromosome goes down. It's not that they all go in the same direction. Crossing over, then, we looked at, this is when you trade pieces of the chromosomes so that you end up with sort of a unique little genetic region there at the end that wasn't present on that chromosome type prior to prophase 1 or metaphase 1. And so you can have, you know, in the end, when the chromatids rip apart, you can have, you know, a mom chromatid with a little piece of dad gene at the end. Fertilization, hopefully, obviously, combines gametes from two different parents. So it mixes genes from two individuals to make unique variations. It's like, you know, shuffling up or remixing of the genes. Obviously, you don't produce identical individuals like asexual reproduction does, because you're combining from more than one source. This is just a look at explaining crossing over again, and we've looked at that pretty thoroughly at this point. Now, random assortment, what you can do is you can follow here. They're trying to show you paternal or father chromosomes in blue and maternal chromosomes in pink. And what you can see is you can end up in, it can happen sort of any way, where you can end up with cells that have all dad's genes in one and all mom's genes in another, or you can have a mix. Realize that humans, down in these haploid cells, for humans, they're actually going to have 23 chromosomes down in these haploid cells, okay? And so all different kinds of combinations, from 1 to 22, you know, 12 and 11, all over with these combinations for random assortment. 23 chromosomes would be lined up in those cells down there. Now, fertilization, you know, hopefully very obviously takes in it, so it'll take a chromosome through the process, divide it out, and then it will send it to a new individual and link it up with another chromosome that just traveled through the process from a different parental individual. And so you're getting a unique combination that wasn't present at the beginning for either individual. And that's what leads to variation, equals new traits or new combination of traits, okay? And this is the basis for, you know, adaptation and natural selection and things like that, is this combination of genes from more than one individual.